In the previous video, I accidentally said that we are going to study about transcription, and I'm sorry for that. I actually meant we are going to study now about translation, and the process which we were discussing is called transcription, or transcribed, or rewritten. So, we left off where we had an mRNA molecule with a cap and a tail ready to go out of the cytoplasm and go into the ribosomes, or where it can be translated. Transcription already occurred. Now we're talking about translation. Transcription occurred. So what happens is that after it leaves this molecule right here, if you want to be specific enough, we call it an mRNA molecule or a messenger RNA molecule. So it leaves the cytoplasm, and it in order, and there's another kind of RNA which I want to introduce to you, is that there are basically two more. One is called the tRNA molecule, and the other one is called an mRNA molecule. Oh, sorry, not an mRNA, the rRNA molecule. So one is called the tRNA molecule, and another one is called the rRNA molecule. So these two molecules are responsible for the translating. Remember, translating. I'm sorry for that. The first part. These are responsible for translating the mRNA into a protein, and the mRNA we got in the result of transcription, and it was transcribed from DNA. So sometimes I get confused between the names. So a tRNA molecule, just for the sake of simplification, has basically two regions: a binding site and and the second part which it has is called the anticodon. Now, an anticodon and a binding site. So if I were to draw a simple structure of this, if this was and this is not actually the structure, I'm just drawing it. If it has this portion right here, I'm just drawing a random shape. This portion right here is the anticodon, this one, and this portion right here is the binding site. Now this is the part of this binding site is the part where the particular kinds of amino acids comes and attach, and you might be thinking, okay, where does the cell gets its amino acid? It either gets it from different kinds of chemical processes, or from the foods we eat, or from the sources of energy we get, such as different kinds of food we eat. So this is one source of getting the amino acids, and this anticodon site has the opposite base pairs, which can actually translate the information. So for example, if this part right here comes along with this, if this part is UCG, its opposite would be A, G, and C. So you can imagine when this part of the mRNA goes through this tRNA molecule, and if this anticodon matches this part right here, it would eventually know that, okay, this information encodes for the specific kind of protein which is attached on my binding site. And this is actually a very amazing process of how it does that. So this one was for the tRNA. And remember, if we have 20 kinds of amino acids, we may have 20 types of tRNA molecules, which each with their own kinds of amino acids on them, with their own kinds of anticodons. And if an amino acid wants to bind to the binding site, it has to go through a chemical process, and it actually occurs with the help of an enzyme, and in that process you use ATP molecule. Remember, ATP is the source of our energy. It's an energy molecule, which is called the currency of energy. Now, so this one was for only the tRNA molecule, and if we are talking about an rRNA molecule, R stands for ribosomes. Ribosomes are the site, or are the protein factories where actually this mRNA and the mRNA goes through the ribosomes, and with the help of tRNA molecules we are able to translate those mRNA molecules mRNA strand into a specific kinds of protein, and it basically contains two different kinds of sites. One is called the larger subunit, and the other one is called the smaller subunit. Oh, this one's becoming more larger. So one is called the smaller subunit, and the other one is called the another one is called the larger subunit. So let me remove this. Now, so if I'm talking about this is the smaller subunit. So this is the larger, and this is the smaller. And the mRNA goes through this, and as it leaves out, the proteins, the amino acid molecule starts building up over here. Now, among this, one part has the P site, and there's a particular place, and another place in the mRNA and the rRNA is called the R site. The P site is where, and the P site and the A site, this place is called the P site, and this one is called the A site. And these two sites are the places where the tRNA molecule comes and attaches itself. So now that we know about different kinds of RNA, let's just go into the process of translation. And I'm again sorry for that. In the previous video I actually switched them up. And just for the reminder, this we got from the process of transcription, and now we're going to study about translation. Now, so now what happens is that if I were to draw a bigger ribosome, now what happens is that the mRNA molecule goes through as the mRNA molecule comes, and it goes through this particular place, which it has a little space for the mRNA molecule to go through, and as it's going through, it's leaving this direction. It's going from here to here. So as it's going through this direction, there is also an A site and there's a P site. So now the whole strand of DNA doesn't encode for us for the protein. So the starting point is, I think it's A, U, and G. So now when the mRNA goes through the ribosomes, and when it goes through this ribosomes, and when the ribosome discovers, oh, here the AUG is right here, so it starts encoding protein from here, from the AUG. If you have AUG here, it starts encoding of protein. Not until, you can see that this part of the protein has already left, but the process doesn't. This part of the RNA has already left, but the encoding doesn't start until the AUG comes along. So as the AUG comes along, which means it starts the encoding of proteins. And what happens is that a tRNA molecule comes along with its anticodon, which if it's AUG, you have U, A, and C. The anticodon of AUG would be U, A, and C. Yes. So it comes here, attaches itself, and it has a specific kind of amino acid attached into it already, and it comes here and attaches itself here. So now this process begins. So now in the P site, this P site has this tRNA. Now as it's going, it has different kinds of sequences all the way. So now these all will be encoded in the protein. This all will be encoded in specific kind of protein. So now another tRNA molecule comes along with its amino acids, and if you have UAC, this anticodon should be A, U, and G, and this molecule with this anticodon comes along to the A site and attaches itself over here. Now this is starting to get a little bit messy. Now what happens is that this bond between this binding site and this protein breaks off. It gives the it gives this amino acid to this tRNA molecule, and this tRNA molecule right here leaves the ribosome. And after it leaves the ribosome, so this tRNA molecule, which now has two amino acids attached to it, it comes and switch its places. It comes to the P site. So the next step would be, if this is the ribosome, and so this one leaves. So if I were to draw, this one is going away, which is this one right here. This one is going away, and this one is coming to the P site, and again the A site is empty. So now you have a tRNA molecule with two amino acids attached to it. Now, so you can imagine, this mRNA strand is just leaving, is just going like through this space right here. So, and if you were to see that there is another kind of code, there's another kind of codon which needs to be translated, and what happens is another tRNA molecule with the opposite of this comes along and joins here, and with its amino acid attached to it. And now what happens is that these two amino acids leave this tRNA and attach themselves to the other tRNA, and now again this one leaves, and this tRNA molecule comes and takes its place. So it leaves the A side and it comes to the P side. So this P side is the place where the whole polynucleotide is supported, or it's the place where the growing polynucleotide is made, and this side is just the place where key amino acids are being added on here. So at the end you end up with a specific codon which says okay, now you have to stop, and as it stops, this polynucleotide leaves and you have a chain of proteins attached to it. So this is what we call translation. So what happens is that a tRNA molecule comes along when he finds that the signal to start the translation is coming, and it comes here, attaches itself to the P side. Another molecule comes along, it attaches itself to the A side. Now the A side has the anticodon of another codon right here, and as it comes here and the other tRNA molecule comes here and attaches itself here, this tRNA molecule leaves. This tRNA molecule causes this amino acid to leave itself and join the other tRNA on the A site, and this tRNA molecule leaves the ribosomes. Now you end up with having two amino acids. So in the same way, this process keeps just on repeating. Another tRNA molecule comes along, attaches itself to the A site. These two amino acids leaves this tRNA which is at the P site and leaves the P site and join themselves to the tRNA molecule which has an amino acid, and then you have three molecules of amino acids were attached to one tRNA, and then this tRNA switches its position and goes to the P site. So this P site is basically the place where the growing polypeptide keeps being supported, and this A site is the place where amino acids are keep being added on here. So hope you find that useful, and bye bye.